Bismillahirrahmanirrahim. First of all, I would like to thank the organizers of this conference. That is the second International Brain and Biomedicines Conference organized by Department of Physiology, Government College University, Faisalabad. I'm Dr. Mahwish Salman, working as Assistant Professor in the Department of Biochemistry, GCUF. The topic of my presentation is the bioprotection of Z maize L by plant growth promoting bacteria against Aspergillus flavus. These are the contents of my presentation. Let's have a brief introduction of my presentation. The plant growth promoting bacteria. These are termed as PGPB or plant growth bacterial species that enhance plant growth and also protect plants from biotic and abiotic stress. These plant growth promoting bacteria also decrease negative environmental effects of chemical fertilizers, pesticides, and microorganisms, and also act as biofertilizer. Pathogenic microorganisms, including various genera of fungus, bacteria, viruses that have adverse effect on maize growth, and they also cause inhibition of plant seed germination. And they also cause a disturbance in physiological and biochemical parameters, reduce yield, and also produce mycotoxins. This is a flow sheet of methodology performed in this project. First of all, uh, we have grown the bacterial species, the bacteria which we used in this study as a treatment is the Cucaria oceani, which is isolated from rhizosporic soil. First of all, we have identified the antimicrobial potential of that Cucaria oceani species. The sequence of that species has already been submitted in NCBI Gene Bank. And then we have collected maize cultivar, that is Melcar 2016, from Ayub Agricultural Research to conduct the pot experiment by bioprime our maize seeds with Cucaria oceani against Aspergillus flavus. After bioprotection experiment of V maize L, we find out the various growth parameters in parallel with various biochemical parameters, including phenolic flavonoid contents, proteins and sugar contents, and antioxidant enzyme measurements. And whereas the growth parameters include root and shoot length and the biomass of root and shoot. These are the different treatments with codes that are applied on maize seedlings. These codes termed as T0, that is the unprimed treatment, means that maize cultivars are not treated with our selected bacteria, that is Cucaria oceani, and even not infected with fungus. These are termed as the controlled seedlings. The second one is T0 bar. It is the treatment named as unprimed seeds of maize infected with Aspergillus flavus. And the third word treatment is T1. That is the maize seeds uh, bioprimed with Cucaria oceani plus infected with Aspergillus flavus. For each treatment, there are three replicates. Three seeds were sown 3 centimeter deep, and after 14 days, samples were collected from pots, and the experiment was performed in completely randomized design. Now we move to the results of our experiment. This is the antimicrobial potential of bacterial strain. Our potential bacteria is Cucaria oceani. As I told earlier, it is extracted and isolated from rhizospheric soil of maize roots. The culture supernatant of Fucaria oceani performed the inhibition zone in millimeter against the pathogenic strains. These are given in the including various bacterial and one of the fungal pathogenic strain, that is the flavus, which is the main infectant in our study. This figure represents the pot, that is the T naught. This one is the center one pot is the T naught, that is the unprimed seedlings plus aspergillus. These are the unprimed seedlings, whereas the T naught bar is the unprimed seedlings plus infected with aspergillus flavus, and this T1 is the bioprimed seeds infected with aspergillus flavus, and you can visualize the effect of bioprimed seeds even in the presence of aspergillus flavus. These are the different growth parameters: root fresh weight and root shoot fresh weight and shoot dry weight. You can visualize distinguishing characteristics between the T naught bar, and T1. Similarly, these are also the growth parameters: the root dry weight, shoot dry weight, and the root length and shoot length. And you can observe that the T1, that is our primed seeds in infected plant. These are the chlorophyll contents: chlorophyll A, chlorophyll B, and total chlorophyll contents. And the same pattern has been observed in these graphs. That is, the minimum chlorophyll contents were found in T naught bar, that is or unprimed seeds infected with aspergillus flavus, whereas the T naught are the unprimed seeds. These are the control, and the T1 are our treated seeds with Fucaria oceani. These are the MDA and hydrogen peroxide contents, and you can visualize that the MDA and the H2O2 contents, these are maximally observed in T naught bar. Whereas the total soluble protein and phenolic contents were found maximum in T1 treatment. Similarly, the antioxidant enzymes have significant increase of the enzymatic activity of SOD, POD, and CAT in T1 treatment. After performing these biological parameters, we have also detected the aflatoxins present in our different treatments: the unprimed control, the unprimed infected with aspergillus flavus, and primed with Fucaria oceani and infected with aspergillus flavus. You can visualize that the AFB1, AFB2, AFG1, and AFG2, no concentration in nanograms per gram was detected in unprimed control, whereas unprimed infected has concentration of these aflatoxins or 4 aflatoxins, whereas the primed seeds with Fucaria oceani and aflatoxins, there no detection of AFB1 and B2 was found. However, AFG1 and a minute quantity or concentration of AFG1 and AFG2 was found. Let's conclude the overall experiment. The current investigations highlighted the use of multifunctional Fucaria oceani to protect maize plant infected with aspergillus flavus by improving photosynthetic pigment, increasing the accumulation of phospholites and antioxidant system of plant. Furthermore, biostimulants reduce oxidative stress in maize plants, reflected by the production of superoxide, hydrogen peroxide, and MDA activity. This work demonstrated synergistic bacterial interactions to explore affordable, eco-friendly biofertilizer for farmers in future. Therefore, field trials are suggested for further studies to understand understanding the potential use of plant beneficial bacteria for sustainable agriculture in fungal infected lands. Thank you so much, and be kind to your mind.